Welcome to the Deep Dive. We're looking at some hot-off-the-press genomics research today, fresh from the ArcSieve server. Our 5 featured papers today all use machine learning and AI to unlock genomic secrets.
We'll cover some really cutting-edge stuff, predicting bacterial traits, generating synthetic DNA, even.
Sounds like a lot to unpack. Let's start with whole genome phenotype prediction with machine learning. Open problems in bacterial genomics. This paper uses machine learning to figure out the genetics behind bacterial traits. Seems pretty straightforward, right?
It's harder than it sounds, like trying to isolate a single flavor in a complex dish. We can predict some phenotypes from genotypes with machine learning, but pinpointing the genes that cause them is a whole other challenge.
So we need to know if a gene is causing a trait, not just associated with it.
Exactly. The paper really digs into the problem of false positives. A model might say a gene is important when it's really not driving the trait. This is especially tricky in bacteria. Their genes are so interconnected.
And why is identifying these causal genes so important? What's the impact?
Think about antibiotic resistance. To develop new antibiotics, we need to know the genetic mechanisms at play. Identifying the specific genes behind resistance could help us design drugs that target them directly.
That makes sense. Okay, on to a paper that's making waves with its ambition. Generator, a long-context generative genomic foundation model. They introduce an AI model that can generate huge DNA sequences, up to 98,000 base pairs.
Longer than a lot of natural genes. And what's incredible is it doesn't just spit out random DNA. Generator can predict the structure of the proteins those sequences would make.
Hold on. It's writing DNA and predicting how that DNA would become a 3D protein.
It is. That opens up some amazing possibilities. Designing new proteins, enzymes that can break down pollutants, antibodies that target specific diseases.
Like a molecular design studio, creating custom proteins for whatever we need.
The potential applications are huge. Personalized medicine, biomanufacturing. But we need to be careful with such a powerful technology. Ethical development and use are essential.
Absolutely. Now for a paper with a more holistic view. Life code. Central dogma modeling with multi-omics sequence unification. This one's all about integrating data from different omics fields to create a bigger picture of biological processes.
Life code is trying to connect genomics, transcriptomics, and proteomics by modeling the flow of information from DNA to RNA to protein, trying to understand a cell's entire production line, from blueprint to final product.
So they're seeing how all these processes work together, not just in isolation.
Right. They use some really cool techniques. A codon tokenizer is one that helps represent genetic information in a way that captures these interactions.
Codon tokenizer. Sounds a bit intimidating.
Think of a codon as a 3-base sequence of DNA or RNA. It codes for a specific amino acid, and amino acids are the building blocks of proteins. The codon tokenizer basically breaks down genetic sequences into these codons, so the model can understand the language of genes on a fundamental level.
So life code sees the codons and can model how they become amino acids, and then how those amino acids fold into proteins.
Precisely. Modeling that whole process from DNA to protein structure could be revolutionary for understanding how genes cause disease and for developing better treatments.
Like a roadmap showing how one tiny DNA error can create a malfunctioning protein and lead to disease.
And life code can trace that path and maybe even help us find ways to correct it.
Our next paper is all about spatial gene expression. Causal GD, blending causality and diffusion for spatial gene expression generation. We know that genes express differently based on where they are in tissues and organs, but modeling that spatial distribution is really hard.
What's cool about causal GD is it doesn't just map where genes are expressed. It tries to understand the causal relationships between them. It takes into account that one gene's activity can directly affect its neighbors.
So it's not just where, but why. Why are genes expressed differently in different parts of a tissue?
Exactly. Causal GD uses that causal information in its predictions, so we get more accurate models of spatial gene expression.
Sounds like a big leap. What could we do with a model like that?
Tons of possibilities. Understanding spatial gene expression is key for studying tissue development, disease progression, and how drugs work. Imagine being able to predict how a cancer drug affects different parts of a tumor. That could help us create more personalized treatments.
Like a 3D map of gene activity, showing us where genes are and how they're interacting?
Exactly. That level of detail could change how we understand tissues, both in healthy and diseased states.
Okay, ready for our last paper today. This one's about interpreting artificial neural networks. They're becoming really popular in genomics, but they can be a bit of a black box.
The paper's called Interpreting Artificial Neural Networks to Detect Genome-Wide Association Signals for Complex Traits, and it's about using neural networks to find genetic variants linked to complex traits. like schizophrenia. These networks are powerful, but it can be hard to know how they reach their conclusions.
Like a detective who solves the case but can't explain how.
Exactly. This paper gives us ways to make these neural networks more transparent so researchers can pinpoint the genetic features the network is using and get statistical significance for those features.
Like giving that detective a way to present their evidence clearly.
Right. That's critical for trusting the results and for understanding the biology behind complex traits.
We've covered a lot today, from predicting bacterial traits to generating synthetic DNA and interpreting those neural networks.
It's a testament to how fast genomics is moving and how much machine learning and AI are driving that progress.
And it's just the beginning. Join us for part 2 of our deep dive. We'll go even deeper into these papers and talk about what they mean for the future.
See you then. Welcome back to the deep dive. Let's jump back into that whole genome phenotype prediction paper. They spent a lot of time discussing false positives in bacterial genomics, a big problem.
Right, it's not easy to find the exact genes that make a bacterium resistant to antibiotics. You can get fooled by genes that look linked but aren't really the cause.
It's like a red herring in a detective story. Seems important at first, but leads you astray. The authors argue that we need to go beyond just correlations and find methods that use biological knowledge and causal inference.
So how do we get AI to understand the biology behind antibiotic resistance? Those processes are complex.
We need to integrate more data. Gene expression patterns, protein interactions. That gives us a fuller picture of how genes function in bacteria.
Like putting together a puzzle. You need all the pieces and to know how they connect.
Exactly. And we need AI that can see those connections too, distinguish between things that are just correlated and things that are actually causing the outcome.
Let's talk more about generative, that AI that makes those crazy long DNA sequences. It still blows my mind that it can make the sequences and then predict the protein structure too.
It's a huge accomplishment. Generator was trained on a massive data set of eukaryotic DNA. It learned the language of life, basically, so it can generate new sequences that look like the real deal.
Like an AI composer, taking inspiration from the classics and writing new music.
Great analogy, but instead of notes and melodies, it's using DNA bases and amino acids, the building blocks of life itself.
And the implications are huge. Like generator could revolutionize protein engineering. We could design all kinds of new proteins, enzymes, antibodies, you name it.
The potential is vast. Imagine proteins that eat plastic waste, or new biofuels, or personalized medicine tailored to your specific genes.
Incredible. But we have to be responsible with this kind of power, Kate.
Absolutely. We need to think carefully about the ethical side of things and make sure these technologies are developed and used responsibly.
Now onto that life code paper, the one that emphasizes integration across the omics fields. It seems like that's a big theme, bringing together different areas to understand the whole system.
It is. Life code is trying to model the flow of information from DNA to RNA to protein to show how those processes are all connected.
Like a cell is a bustling city. All the different departments have to work together.
And life code is like the city planner, mapping out how everything interacts.
How do they actually do that? You mentioned something called a codon tokenizer.
Right, so a codon is a 3-base sequence in DNA or RNA. It codes for a specific amino acid. And the codon tokenizer breaks down genetic sequences into those codons, so the AI can understand the language of genes in a much deeper way.
Kind of like translating a complex text. You have to keep the meaning in context.
Exactly. By understanding codons, life code can model the whole gene expression process, transcription, translation, protein folding.
And that could help us figure out how genes cause disease and maybe even find better treatments.
Right. Imagine being able to pinpoint the exact molecular error that's causing a disease and then designing a drug to fix it.
Like a repair manual for the human body.
Exactly. Life code is a step towards that kind of precision medicine.
Let's move on to that paper about spatial gene expression, causality. They look at how gene expression changes depending on location in a tissue.
And what makes causality unique is it tries to figure out the cause and effect relationships between genes in different parts of the tissue.
So it's not just mapping where genes are expressed, but also understanding how one gene's activity might affect its neighbors.
Exactly. Gene expression is dynamic. There's constant interaction. And causality is trying to capture that complexity.
Like a neighborhood. What one person does can affect everyone around them.
And causality can model those ripple effects to create a more accurate picture of what's happening in the tissue.
What kind of benefits could that have, understanding those causal relationships?
It could change how we think about tissue development, disease progression, drug response. Imagine predicting how a cancer drug will affect gene expression in different parts of a tumor. That could lead to more targeted treatments.
It's like having a 3D map of a city, not just showing the buildings, but how the traffic flows between them.
And if you understand those traffic patterns, you can optimize things, from transportation to urban planning.
Finally, let's touch on that last paper about interpreting those artificial neural networks.
Right, the one about genome-wide association studies, they're tackling the problem of understanding how these complex neural networks work, because they're powerful but often opaque.
Like our detective who can't explain their reasoning.
Exactly, but this paper gives us tools to make those networks more transparent so researchers can see what features the network is using and get statistical support for their findings.
Like giving our detective a way to show their work so we can understand their conclusions.
Right, that transparency is key for building trust in the results and for figuring out the biological mechanisms at play.
Back again for the deep dive. We've spent the last 2 parts looking at these 5 incredible papers, all about how machine learning and AI are changing genomics research.
We've seen how they're being used to predict bacterial traits, generate synthetic DNA, integrate multi-omics data, model spatial gene expression. We even talked about how to interpret those complex neural networks.
It's been a wild ride through some cutting-edge research. Now it's time to step back and think about what we've learned. What are the big takeaways here? And what do these advances mean for the future?
One thing that really stands out is the speed of innovation in genomics, and that's largely driven by this fusion of biology and computation.
It's like those 2 fields have finally found a way to talk to each other.
And they're doing some amazing things together. We're moving beyond just describing genes and sequences towards a more predictive and even generative genomics.
Like we've gone from reading the book of life to writing new chapters.
And the tools we're using are getting more and more powerful. Now we have machine learning models that can analyze huge amounts of genetic data, but they can also generate new sequences and predict what those sequences will do.
Generative is a great example. That AI that can make synthetic DNA. It's incredible how accurate and long those sequences are.
It shows us what generative AI can do. It can mimic nature, but it can also go beyond nature. We could use models like that to design proteins that do specific things. The applications are endless. Medicine, biomanufacturing, you name it.
It's like we have a molecular design studio at our fingertips.
But we have to be careful with this power. It's important to think about the ethical implications and make sure these technologies are developed and used responsibly.
Absolutely. Another big takeaway is integration. We can't keep studying biological processes in isolation. We need to connect the different omics fields to see the bigger picture.
That LifeCode paper was a good example of that. They integrated data from genomics, transcriptomics, and proteomics, so they could model the entire flow of information, from DNA to RNA to protein. That gives you a much more complete understanding of how genes work.
It's like putting together a puzzle. You need all the pieces to see the whole picture.
And that integrative approach is key. It helps us understand how biological systems work. And it can lead to better diagnostics and treatments for diseases.
We also saw how machine learning can help us interpret those black box neural networks, which are becoming so common in genomics.
That paper about genome-wide association studies, they were trying to make those neural networks more transparent so we can understand how they reach their conclusions.
Like making that detective show their work.
Right. That transparency builds trust, and it helps us figure out the biology behind these complex traits.
It's clear that innovation in genomics is just going to keep accelerating. We'll see even more powerful machine learning models, more sophisticated ways to integrate data, and more amazing applications in healthcare, biotech, all kinds of fields.
Imagine a future where we can predict your risk of disease based on your genes, or design personalized therapies just for you, or engineer organisms to solve environmental problems.
Sounds like science fiction.
It's getting closer all the time, but as we go forward, we need to use these powerful technologies wisely to benefit everyone.
It's a responsibility we all share. Scientists, citizens, everyone.
Thanks for joining us on this deep dive into genomics. We hope you learned something new and that you're excited about the future of this field.
Until next time, keep exploring, stay curious. And remember, the future of genomics is in our hands. 3500.